Great. All right, welcome everyone. It is my great pleasure to introduce Dr. Ben Stanger. Ben Stanger is the Hannah Wise Professor in Cancer Research at the University of Pennsylvania and Director of the Penn Pancreatic Cancer Research Center at the Abraham Family Cancer Research Institute at the Perelman School of Medicine. So Dr. Stanger has really made some very seminal discoveries in pancreatic cancer. I'll just describe a few here. Early on, he introduced the fluorescent marker YFP into a mouse model of spontaneous pancreatic cancer and used this to make some really surprising discoveries that cancer cells, or really epithelial cells, disseminated very early, even from pre-neoplastic lesions. He's gone on to show that the circulating epithelial cells can be found in patients with pancreatic precursor lesions as well, and has looked very extensively at the early stages of oncogenesis in mouse models, characterizing tumor cell intrinsic mechanisms of EMT and transition to metastasis. More recently, his work has moved to understanding the immune and stromal factors that also contribute to early metastasis, both in the primary pancreas and also in distant sites of micrometastatic disease. In collaboration with Bob Vanderheide, his lab showed what is, in my opinion, still the most definitive evidence that tumor cells orchestrate their own microenvironment by producing a really Herculean library of clonal cell lines that differed in their ability to exclude CD8 T cells. These T cell high and T cell low models really set a new standard in the field of pancreatic cancer research and give one illustration of how Dr. Stanger's scientific rigor continues to elevate the entire field. So we're very much looking forward to his talk today, and I will pass it over to you.
Okay, well, thank you so much for that introduction, Stephanie. I am delighted to be here, and I'm going to share my screen. No, I don't want to share it that way. Share my screen this way. Why is it doing this to me? I'm going to try this one more time.
Great, and as we're getting the slides up, if people have questions during the talk, you can go ahead and type those into the Q&A session, and then we'll come to them at the end.
All right, are you able to see this in the correct mode, or is this in?
No, we see it in presenter mode.
Backwards mode. Oh no, I thought we had this all worked out before, but something's different now. I apologize for this. I'm so sorry about this. Let me pull this up again.
All okay.
Try resharing and just go into presentation mode at the bottom. Right, and then go to presentation mode at the bottom.
Okay.
There
Does
you
this
go.
look, this looks good
That's
for you?
perfect.
That's fine.
All
Yes.
right. All right. So sorry about that. Well, thank you, Stephanie, for that lovely introduction. I'm really happy to be here virtually. I wish I could be there in person. Thank you for joining on this very frigid Boston and also cold Philadelphia day. These are my disclosures, none of which are relevant for the talk today. So today the topic is cellular plasticity. And what I mean by cellular plasticity is a reversible change in cellular phenotype without any alteration in the genotype. So there's nothing really magical or mysterious about cell plasticity. It's a feature of normal development. During development, cells change their phenotypes. That's part of normal differentiation and the ability to change a differentiation pathway should anything abnormal happen during development. And plasticity is not really a feature of adult tissues normally, but it does accompany injury and regeneration. And cancers frequently exploit plasticity during tumor evolution, and that allows them to cope with selective pressures and overcome therapies in some cases. And also it creates quite a bit of heterogeneity and diversity, which cancer cells can exploit as they evolve. And for those who don't know, this is an image from one of the Terminator movies where the Terminator's cells are exhibiting plasticity so that he can walk through walls. All right, so this is the outline of the talk. Today, I'm going to give a brief introduction to pancreas cancer and the models that we've been using in the lab. And then really, I'm going to tell two stories. The first is about epithelial mesenchymal plasticity, which is related to metastasis, and much of this is published. And then the second story will be about metabolic plasticity, and this is all unpublished work that I'll be describing. All right, so for introduction, I think most of you are probably aware that pancreas cancer is a bad disease. And I think this graph of data from the UK illustrates that and illustrates how far we have to go. So these are 5-year survival curves for various tumors from the 1970s to 2015 in the UK. And there are two take-homes from this graph, I think. The first is that for most tumor types, there's been really quite a bit of progress. And I think we've all seen that. And there it is by the numbers. And I think this is similar in the US. But if you look at the very bottom here, you can see pancreas cancer. It really has not benefited from the advances in precision medicine, in immunotherapy and other types of therapy that other tumor types have. So pancreas cancer is currently the third leading cause of cancer death in the US. It's predicted to become the second over the next few years. Median survival, even given best therapy, is under a year. And there are a couple of reasons for this. One is that most tumors have already spread at the time of detection. So metastasis is a big part of the problem, and the lack of effective therapies. Combination chemotherapy has had some impact, but we really don't have a good arsenal for therapy for pancreas cancer the way we do for these other tumor types. We do know a lot about the biology of this tumor, however, and so this is one illustration of the sort of standard dysplasia to carcinoma sequence that was first described in colon cancer, but is more or less true in every solid tumor. So the dysplastic lesions here, the equivalent of adenomas in the colon, are called Pannin lesions for pancreatic intraepithelial neoplasia. They have various degrees or grades, and at some point they transition into an invasive carcinoma that can disseminate and metastasize. And by studying these lesions over many years, researchers in the field were able to correlate these histologic events with molecular events, principally mutations in 4 genes, the so-called big 4 of pancreas cancer, because all other mutations happen with a much lower frequency than these 4 genes. And they are KRAS, CDKN2A and 2B, P53, and SMAD4. So all of this information, the histology, the molecular information, a number of years ago allowed several models, mouse models, of pancreas cancer to be developed. Principal models were developed by Dave Tuvison and Sunil Hingorani. Andy Aguirre, who may be in the audience, was responsible for developing another model at the Farber. And what these models use is those signature mutations. In this case, the so-called KPC model uses a KRAS, a mutant KRAS, knocked into the KRAS locus, a mutant copy or a deleted copy of P53, and a Cre recombinase that's directed specifically to epithelial cells in the pancreas. And when all 3 of these alleles are present in the same animal, the animals develop a Pannin to pancreas cancer progression. So what we did a number of years ago, and Stephanie mentioned this in the introduction, is we added a lineage trait, so YFP allele, to this combination. And that resulted in the epithelial cells of the pancreas being labeled with a irreversible fluorescent marker from the onset of development at the same time as this mutant KRAS and mutant p53 were being generated. So that made the entire pancreas appear green, fluorescently labeled. But it also allowed us to do a number of things as we were studying the biology of the tumor. So it allowed us to distinguish the epithelially derived cancer cells from cells that were not derived from the epithelium, so the stroma. This allowed us to study EMT, which is going to be a focus of what I'm talking about today, because cancer cells, even if they no longer look epithelial, they've undergone a transition to a more mesenchymal state, they'll still carry that lineage label, so we'll be able to identify them. And I'll get into that more in a bit. This has also allowed us to study circulating tumor cells because we can detect them based on their fluorescence. And we can study the process of colonization, how cells, once they've made it to a distant organ, are able to grow out, even at single cell resolution, because of this fluorescent marker. So what's shown here is a section from one of these KPCY animals stained with E-cadherin, an epithelial marker, and DAPI. And so you can see in the tumor these glandular structures that we would easily be able to recognize as tumor cells. And everything out here we would consider to be the stroma, the leukocytes and endothelial cells and fibroblasts that make up the tumor microenvironment. But the power of the lineage trace is that it allows us to see cells that have maybe lost some of their epithelial features, but are hanging out in the stroma that we can now detect by virtue of this YFP lineage label. This is a rather egregious example of this kind of EMT phenomenon, but it sort of illustrates the point that this allows us to see cells we wouldn't otherwise be able to detect. So just a couple of slides to show you this model in action. Here we've taken blood from a tumor-bearing animal, KPCY animal, stained for CD45, and we're, for this purpose, not interested in the blood cells, but we've stained for YFP, which we know are going to be tumor cells, and we can find those tumor cells relatively easily in the blood. So if we quantify that, we can find on average around 100 of these circulating YFP cells in a KPCY mouse at any point in time. Lineage traced animals that have a green pancreas, but no oncogenic mutations, really don't have any cells in their circulation as you'd expect. One of the surprises that was mentioned in the introduction is that if we look at animals with Pannin lesions, so that is to say we look through the pancreas, we don't see any evidence of invasion, pathologists wouldn't see that this is a carcinoma, we can actually still see some of these cells that have made their way out into the circulation. They're present in lower numbers, but they're still there. And in fact, they're not just in the circulation. We can actually find them in distant organs, such as the liver, where they are in sinusoidal endothelia vessels, or in some cases, having left the vessel and living in the parenchyma of the distant tissue itself. So this maybe isn't surprising, given what I told you early on, that we know that pancreatic tumors disseminate very early. They've almost all metastasized at the time they come to clinical attention. But here we can see it directly in this mouse model. And it sort of highlights the point that we can't always tell what's going on from histologic criteria alone. These are animals that, again, we can't see evidence that the cells have invaded, and yet the cells are capable of making it all the way out throughout the body. This is just a slide or two to illustrate how we can use this to study metastasis. So here we can take an animal that has a primary tumor and look through its liver, and in any given animal we can find lesions that span the entire spectrum of colonization, from lesions that are gross metastases that you could recognize by eye, all the way down to single cells and everything in between, micromets, which we've just defined as having 11 to 100 cells roughly, and nanomets, which have anywhere from 2 to 10 cells. And by characterizing all of those different size lesions, we can make interesting observations just about what happens as a metastatic lesion grows. So here we're looking at blood vessel density. And what we see is that cells that have made their way to the liver as single cells, they're right in the liver parenchyma, they're surrounded by all of the sinusoidal endothelium of the liver, but that as the lesions grow, they develop a desmoplastic or fibroblast-rich stroma that creates this sort of halo effect. So the tumors, pancreatic tumors in general, tend to be hypovascular because of all the fibroblasts and matrix that they present. And over time, they come to resemble the primary tumor, which is hypovascular. And this sort of triggered us to look in human pancreas cancer, where we could compare small and large liver mets to primary tumors, and we saw essentially the same trend. So that essentially just gives you a flavor for pancreas cancer in the models, and now I'll launch into how we've used these models to study epithelial mesenchymal plasticity and metastasis. And the work I'm describing now is the work of 3 graduate students in the lab, Nicole Aiello, Bobby Norgard, and Selena Yuan. So a little bit more about epithelial mesenchymal plasticity, or EMT and MET. This is a transition between cells in an epithelial state, forming an epithelium with all of their tight junctions and gap junctions here, to cells with a more mesenchymal phenotype, like a fibroblast or a leukocyte. Features of this, this change, this plasticity, is that it's phenotypic. It occurs along a continuum, cells of all intermediate stages can be found. There are a variety of markers that can be used to distinguish cells in either an epithelial or a mesenchymal state. None of them are perfect, and all of them differ to varying degrees in different models as cells are undergoing this plasticity spectrum. So it can be hard to use markers to understand exactly what's going on. MET and EMT are associated with different drug sensitivity. So a cell in a more mesenchymal state may have a different vulnerability to certain agents than when it's in the epithelial state. And this provides some opportunities for precision therapy, although that hasn't really been borne out yet, but in principle it should. An important feature of epithelial mesenchymal plasticity is that cancers haven't invented this. This is borrowed from embryonic development, and 2 examples of how this is critical and important for development is neural crest migration, where cells from the neural tube undergo an EMT and are able to disseminate throughout the body, giving rise to melanocytes and cells in the heart, etc. And it's also critical for gastrulation, where cells that are part of the epithelial epiblast break away and again give rise to all of the germ layers that make up our bodies. So in principle, the same processes and phenomena that give rise to EMT in the embryo are the ones that cancer cells are probably latching on to, and I'll try to touch on that later. All right, what is our current conception of EMT? We think of EMT largely, or have traditionally thought of EMT as a transcriptionally regulated process. This is a busy diagram from a recent review showing that there are a series of so-called EMT transcription factors, or EMTTFs. They go by the names ZEB, SNAIL, PRX, TWIST. And what these do collectively is transcriptionally repress genes that are involved in the epithelial phenotype and activate genes that are involved in the mesenchymal phenotype. So what I'm showing you here is example of this from our lab. This is traditionally studied in cancer, at least, in cell culture with cell lines, where one can induce cells to undergo an EMT by giving them TGF-beta, or other factors can do this as well. And you can see this dramatic change in cell phenotype. And there's a transcriptional induction of these EMT transcription factors and other mesenchyme-associated genes, and a transcriptional repression of epithelial genes like E-cadherin, claudins, etc. And this type of experiment has been repeated hundreds or thousands of times and is really the paradigm. And of course, these changes in E-cadherin mRNA are reflected in changes at the protein level as well when induced in this kind of a culture setting. The reason, of course, we're interested in EMT is its relationship to metastasis. This change in cell phenotype enables cells to break away from their epithelial layer, which by definition is required for a cell to be able to invade. But there are some puzzling things about how EMT is involved in metastasis, which is what really prompted our interest in it. What I'm showing you here is an overview of what's called the metastatic cascade, all of the different steps that a cell has to go through in order to disseminate to a different site. First, it has to break away from the primary tumor, get into the bloodstream. It has to survive the stresses of the bloodstream, anoicus, shear stress, immune attack. It has to get out of the bloodstream or the lymphatics, and then it has to grow at that distant site. And each of these are relatively infrequent events. So metastasis actually, cells have to go through a number of hurdles to be able to metastasize. What's interesting about the invasion step is that there are two basic classes of migration that have been observed in solid tumors. And one of them is called collective migration, where you can see a so-called invasive front. The cells are moving together as a group. The other is single cell invasion, where you can see single cancer cells moving separately. Another difference that one can see in if one looks at the bloodstream is that sometimes tumor cells travel as single circulating tumor cells or CTCs, and sometimes they travel in the bloodstream as CTC clusters. So all of these different phenomena raise a number of questions. How is EMT involved in cancer cell dissemination? Why do cells spread as single cells sometimes, as clusters as other times? Is collective migration an EMT independent way of invading? So these are the kinds of questions that we were interested in. We felt like we had a good model for looking at this. So this was the project that Nicole took on as a graduate student. I've shown you this picture before where we can nicely see cancer cells because they're labeled with this YFP lineage trace, but we can separate out those that have an epithelial phenotype by the presence of a surface E-cadherin molecule, and those that have lost their epithelial phenotype because they no longer express E-cadherin on the surface. And what Nicole did is she sorted first the tumor cells that express the YFP, and then further sorted them based on their E-cadherin surface expression into those that had an epithelial phenotype and those over here that had lost their epithelial phenotype. And then she did RNA sequencing with the expectation that we would see all of these cells across multiple tumors fall into two groups. The epithelial cells and the mesenchymal cells would be different based on the repression of all of the epithelial genes at a transcript level. But instead, we saw something different. We saw that the tumors actually clustered into two groups rather than the cells clustering. So here are the two groups, and what I'm showing you here is a heat map where we're looking at the relative expression of a number of genes, epithelial genes, mesenchymal genes, or collagens, in the ECAD negative cells of the tumor compared to the ECAD positive cells of the tumor. So in this group of tumors here in cluster 1, we can see that they behave exactly as the paradigm would have predicted. There's a transcriptional repression of all of these epithelial genes. But in cluster number 2, these cells that are E-cadherin negative at the surface still have relatively normal levels of expression of all of these epithelial genes, E-cadherin, EpCAM, claudins, epithelial keratins, etc. And in both categories, cluster 1 and cluster 2, there's an upregulation of a variety of mesenchymal genes and collagens. So if this might be a little confusing, I'll just boil it down to 3 epithelial genes here, E-cadherin, EpCAM, and claudin. So in cluster number 1 tumors, which we call classical EMT, there's a nice transcriptional repression of E-cadherin, EpCAM, and claudin in the cells that are ECAD negative. But in the cluster 2 tumors, which we call partial EMT, you can see that even though the cells are negative for surface ECAD, they have normal levels of ECAD, EpCAM, and claudin 7 transcripts. And so what this must mean is that in the majority of the tumors, these cluster 2 tumors, E-cadherin has been lost as a result of a post-transcriptional event rather than transcriptional repression different from the paradigm. So there could be a lot of reasons for this post-transcriptional event. It could be that the protein isn't translated. It could be that it's translated and degraded. The answer turns out to be that it is relocalized. So we made a number of cell lines that recapitulate this classical EMT or CEMT versus partial EMT, PEMT phenotype. And in the CEMT cells, if we sort the cells based on their E-cadherin expression, we can see that in the ECAD negative cells, there's absolutely no ECAD protein. There's no transcript nor protein. But in these partial EMT cell lines, even in the ECAD negative cells, by sorting, there's abundant ECAD protein, so it's not being degraded, it's being translated. But if we look where it is, it's actually inside the cell. It's trapped in these recycling endosomes that express Rab11. And E-cadherin is normally subject to lots of trafficking and recycling from the membrane back onto the surface. And in these cells that have undergone a partial EMT, something has interrupted that return to the surface. And this is, I'm only showing you E-cadherin here, but this is true of a number of different epithelial proteins, such as beta-catenin and other components of the adherence junction. This is another way of looking at that. So here we do a two-step staining protocol where we first stain the cells for surface E-cadherin and then permeabilize and use a different antibody to stain for intracellular E-cadherin. And here what we're mapping across different cell lines is the percentage of cells that are negative for surface E-cadherin that have intracellular E-cadherin. So in the classical EMT tumors, any cell that lacks surface E-cadherin also lacks E-cadherin inside the cell. There's no RNA, there's really no protein anywhere. But in the partial EMT tumors, the majority of cells that have lost their surface E-cadherin still have stainable E-cadherin inside the cell. Okay, so how does this relate to the metastatic cascade and these questions that I raised earlier? First of all, we wondered whether in this model we could see both modes of invasion, single cell invasion and collective invasion, or single cell CTCs and CTC clusters. So Ravi Madhupati, a former postdoc in the lab, took a number of KPC animals and looked at the blood and then asked what percentage of tumor cells that express a YFP in the blood are present as single cells or as clusters. And just by looking at a number of animals, roughly 10 to 20% of the cells that travel in the blood are present as clusters rather than as single cells. So clusters do emerge spontaneously in this model. In addition, we found that clusters were far more capable of generating metastases than the single cells. This had already been reported by Dan Haber in the MGH group, but we repeated that by making clusters of cells and showing that their ability to generate metastases was much, much higher than single cells. So then we took our lines that recapitulated either this CEMT phenotype or the PEMT phenotype. We made clusters and embedded them in Matrigel and just asked, by making movies, how do they seem to invade into the Matrigel? And what we found was that the CEMT cells generally tended to invade as single cells, whereas the PEMT cells had a much higher chance of breaking away from the cluster, from the main cluster as a cluster of invading cells. So if we then take these cell lines, the CMT cell lines and the PEMT cell lines, and embed them into mice and make tumors from them and allow those tumors to now invade into the bloodstream and look at the bloodstream, this in vitro phenotype is recapitulated. The CEMT tumors give rise to a large number of single CTCs in the bloodstream, but very few clusters, whereas the PEMT cell lines gave rise to a large number of clusters in addition to being able to give rise to single CTCs in the bloodstream. And if we look at what is present in the bloodstream, the CTCs that come from the The classical EMT tumors, the ones that have transcriptionally repressed their epithelial program, there's really no E-cadherin, and all of these epithelial proteins are gone. They're really on their own. But the PEMT tumors that have used this post-transcriptional relocalization mechanism, they still have E-cadherin, and they're capable of putting that E-cadherin on the surface when they see another cancer cell, and thereby form adherence junctions. So the E-cadherin is all localized in these clusters at the site of cell-cell contact, but there's no E-cadherin on the outside. So as far as the bloodstream is concerned, this is a mesenchymal cluster, but there are epithelial characteristics that allow the cells to be bound together. Lastly, I'll just point out that although this was all done in a model of pancreatic cancer, we looked for evidence across other cell lines in breast cancer and in colon cancer in particular to find whether there was evidence of this dichotomy between partial EMT and classical EMT programs. So what I'm showing you here is data from a number of breast cancer cell lines, which we predicted based on signatures to have this partial EMT phenotype. So some of these may be familiar to some of you. And if we then sort the cells into those that are E-cadherin positive or E-cadherin negative by sorting at the protein level, and then look at ECAD RNA, can see that regardless of whether they're ECAD positive or ECAD negative, they have similar levels of ECAD RNA. And then if we take those cell lines and we now do this two-step staining protocol I described to you and ask what percentage of the cells that lack surface E-cadherin have intracellular E-cadherin, it's the majority, whereas in the CEMT cells, they don't have any on their surface, they don't have any inside the cell. They really have undergone a much more complete EMT where they're devoid of these epithelial RNAs and proteins. Okay, so I'm just going to spend a few minutes talking about mechanism and what we've explored before moving on to the second part and metabolic plasticity. So we are still in the process of trying to understand this partial EMT mechanism that's post-translational. We think that it involves a G protein coupled receptor signaling and elevated calcium flux inside the cell, and we've published a little bit on that, but that's work in progress. What I'm going to focus on instead right now is the mechanism of this classical EMT, which is transcriptionally based. And one of the things we wondered was whether this epithelial mesenchymal plasticity, which I told you is regulated by EMT transcription factors, might be regulated by a higher order epigenetic regulatory process, especially since the cells seem to have some memory, some durable memory of whether they're in an epithelial state or a mesenchymal state. So this was the project of Selena Yuan, an MD PhD student in the lab. And Selena conducted a CRISPR screen, which I'll tell you about, but before she did the screen, she wanted to make sure that what she was looking at was really plasticity. And so what I mean by that is you could imagine that there's a cell line and half of the cells are locked in an epithelial state and half the cells are locked in a mesenchymal state. And you see that there's both in the culture, but they're not really moving back and forth as we would want to see if we're really discussing plasticity. So what she did is she took a number of these KPCY cell lines and she sorted on E-cadherin into an epithelial population and a mesenchymal population, ECAD positive, ECAD negative, and then plated the cells. So here, for this cell line, she's plated the ECAD positive population and the ECAD negative population at time point 0, and then watched what happened over time. And what this dotted line is showing here is what the starting percentage of ECAD positive cells was before she did the sorting. And what you can see is over a period of 2 to 4 weeks, the cells returned to that baseline percentage of around 40 or 45% E-cadherin positive. So the ECAD positive cells are becoming ECAD negative, the ECAD negative cells are becoming ECAD positive. There's really a true plasticity. And this is true of other cell lines as well. And what this also tells us is that each cell line has its set point for where the cells want to live in terms of their epithelial mesenchymal state. We're all familiar with some cells that when they're grown in culture have a very epithelial state, but maybe there are some mesenchymal cells in the culture and vice versa. well, if you were to sort those different phenotypes out separately, the cells would in all likelihood return to that basal set point that they exhibited at the initial time point. So this then gave Celine an opportunity to do a CRISPR screen where she introduced a library of around 800 epigenetic regulators and transcription factors into one of these cell lines, allowed the cells to move back and forth between the epithelial state and the mesenchymal state, and did sequencing for the different guide RNAs at various time points. And with the idea that any guides that were enriched in the E-cadherin positive population, the epithelial population, would be genes that were important for establishment or maintenance of the mesenchymal state, and vice versa. Anything enriched in the mesenchymal population would be potentially involved in establishment or maintenance of the epithelial state. Anything such as those genes or guide RNAs represented by the purple bar here that were lost in both would just be genes that were important for cell viability, for example. So this is one illustration of the kind of data that Selena got. She used a number of controls. I'm showing you here ZEB1 controls, which, as we would have predicted, are enriched in the ECAD positive cells because this EMT transcription factor, when it's lost, tends to push cells, allows cells to remain in the epithelial state. E-cadherin itself was a control for the mesenchymal state. And two candidates really jumped out to her. One of them, a gene called NSD2, which is a histone methyltransferase, which was among the most highly enriched genes in the ECAD positive population, really about the same level as the ZEB control. And then another gene, KDM2A, which is histone demethylase, which was also highly enriched, not quite at the same level, but also highly enriched. But the reason that these were of interest is that they both have the same target inside chromatin, and that's histone-free lysine 36, where they add or remove a methyl group. And that lent itself to a very nice model, which is that perhaps the epithelial state epigenetically is regulated by these two genes, where the presence of a dimethyl H3K36 is associated with the mesenchymal state, and a monomethyl is associated with the epithelial state. So she did a number of experiments to validate this. I'll go through them really quickly. The knockout of NSD2 doesn't allow this dimethylated form to exist, so cells become more highly epithelial, and that's illustrated by this E-cadherin staining pattern here. We could also see this when we made tumors from the NSD2 knockout cells, whereas the cells might be very poorly differentiated tumors. In the wild type state, the knockout results in them now being better differentiated and now expressing lots of E-cadherin and other epithelial markers. The model, though, isn't that it's NSD2 that's doing this, it's that the histone mark itself is a marker of the epithelial or mesenchymal state. So here, Selena did a number of correlative experiments. First, of course, to show that NSD2 knockout or KDM2A knockout resulted in the appropriate change in this mark. So NSD2 knockout in a variety of cell lines causes a loss of H3K36 dimethyl, whereas KDM2A knockout causes an increase in the mark. But then if we use other ways of looking at epithelial mesenchymal state, such as TGF-beta treatment results in an increase in the level of this mark. If we sort for spontaneous EMT, so that is to say we sort for cells based on E-cadherin positivity in a standard culture, the cells that have undergone EMT that have lost E-cadherin have higher levels of this mark. And if we do histone mass spectrometry comparing these ECAD positive and ECAD negative cells, the only histone mark that's globally different and enriched in the ECAD negative cells is H3K36 dimethyl, either in the H3.3 gene or H3.1 gene. So to functionally show that it's this mark itself, not any of the other genes that are capable of regulating K36 dimethylation. Selena took advantage of this mutant K36M, which blocks all of the genes capable of dimethylating K36. So now there's no K36 dimethylation, and this results now in the cells becoming much more epithelial because they're now unable to form the dimethyl mark. But in addition, the cells now become resistant to TGF-beta induced EMT. Normally, if you induce EMT with TGF-beta, you see a loss of E-cadherin and EpCAM, and induction of mesenchymal markers. But now, when the cells are prevented from acquiring this H3K36 dimethyl mark, the cells no longer can undergo a TGF-beta induced EMT. Finally, she looked at the regions in the genome that might be governing this. She found that NSD2 deletion resulted in two classes of genes, those genes that are those peaks associated with the loss of the dimethyl mark, and those peaks in which the K36 peaks were retained. So these would be genes where NSD2 loss is associated with the inability to undergo EMT, and these are places where NSD2 isn't active and the mark isn't changing when you lose NSD2. So K36 is being maintained by some other regulator. So when she looked at which genes were associated with these presumably irrelevant K36 peaks or irrelevant to this process, they included genes like E-cadherin and EpCAM, but there was no change in the expression of these genes in and of itself. So, so these are genes that are not being directly regulated by NSD2 deletion or by this mark. However, when she looked at those genes that NSD2 deletion causes a loss of the K36 mark, there were 189 genes that fell into that category. Their transcript levels were lower, and remarkably, almost all of the EMT transcription factors fell into this category. So this really led to a very clear model, which is we've been thinking about EMT as being controlled by these EMT transcription factors, but there's, we think, likely to be a higher level of regulation where those EMT transcription factors have to have the presence of an active enhancer for them to be expressed, and that active enhancer state is marked by H3K36 dimethylation that is under the control of histone writers such as NSD2 and KDM2A. So to summarize the first part of the talk, what I've told you is that epithelial plasticity manifests in vivo through two different programs, a classical or CMT program and a partial PEMT program. The CMT program is governed by both transcription factors and an epigenetic mechanism overriding that, governed by H3K36 dimethyl histone mark. In most tumors, which are dominated by this post-transcriptional mechanism of EMT, we think that G protein coupled receptors and calcium flux are responsible for that. And these different ways that cells undergo EMT enable them to either invade collectively, giving rise to circulating tumor cell clusters, or to invade as single cells, giving rise to single CTCs. All right, in the second part, I'm going to try to speed it up because I've gone a little longer than I wanted. So to talk about metabolic plasticity, this is a work of a postdoc, Yogev Sella, and another graduate student in the lab, Jinyang Li. So it should come as no surprise that solid tumors have gradients of nutrient and oxygen availability. So for example, this is an illustration taken from a review where you can imagine that cells that are close to a vascular supply have access to nutrients and oxygen and all the things that they want and are readily proliferative. But as you move further away from the blood vessel, you enter a quiescent zone where the cells have to live on much lower levels of nutrient and oxygen availability. And when a cell is far enough or long enough removed from any nutrients or oxygen, it's going to be in a very serious trouble, a hypoxic or necrotic zone. So if we look at pancreatic tumors, we can see direct evidence for this. So this is 3 different single-cell RNA-seq data sets where we can curate out the cancer cells and then apply cell cycle signatures to see who's dividing and who's not dividing. And it turns out that about 10 or 15% of the cells in a pancreatic tumor, this is human pancreatic tumor, is dividing. And you can get the same kind of result by doing, for instance, Ki-67 staining, looking at the cancer cells, and you'll find that only a minority of the cancer cells are in the cell cycle at any point in time. We can also see this in the mouse model, where we can actually spatially look at where these cells are located. And so in the mouse model, we inject intravenous dextran into a tumor-bearing mouse. The dextran goes wherever it can perfuse. Where it's far away from a blood vessel, it won't be perfused. And then we can score areas based on the dextran density. We can categorize an area as well perfused or poorly perfused. And as you might expect, cancer cells marked by this YFP lineage trace in the well perfused area are dividing a lot faster than cells in the poorly perfused area. They have a lot more of the goodies that they want. But there really isn't cell death, much cell death in either area. So those cells in the poorly perfused area have figured out a way to get by without dying, even though they're living on the edge in the absence of adequate nutrition. So because Yogev is Israeli, we came up with this just simple term to think about these 2 areas that are nutrient and oxygen deprived, we call arid areas of the tumor microenvironment, and those which have greater access to nutrients and oxygen we call fertile areas. And so what Yogev wanted to do was develop an in vitro system that modeled these arid areas. And the idea was that if they're far removed from blood vessels, they should be deficient in everything. So rather than studying one metabolic deficiency at a time, either oxygen or glucose deprivation or amino acid deprivation, Yogev empirically played around with conditions that sort of depleted everything. And he arrived at this set of conditions, so 1% oxygen, 2.5 millimolar glucose, 0.5% of fetal serum, and 20% of the standard complement of amino acids. And under these conditions, the cells really slowed down their proliferation, but they were completely viable. So we don't know exactly, how well this approximates any given metabolic microenvironment within the tumor, but at least phenotypically, it behaves the way that we would expect it to in an arid part of the tumor. Other phenotypic characteristics that are in line with that, well, under arid conditions, nutrient scavenging goes up. The cells exhibit lots more autophagy. They undergo a process known as macropenocytosis, where they eat the proteins in their microenvironment to a much greater degree. This arid state or nutrient and oxygen deprived state renders the cells quiescent, and that also has the effect of rendering them resistant to therapies that only act on dividing cells. So gemcitabine, one of the standard therapies for pancreatic cancer, is quite effective at killing cells at higher doses under fertile conditions when you have the cells dividing rapidly. But under these arid conditions where the cells are quiescent, you can add millimolar concentrations of the drug and the cells are perfectly happy, they don't die. And this is also reversible. So if we use a dye that gets diluted as cells divide, we can see that cells, this is doubling times, cells that are in arid conditions for a week or 3 weeks, double every 6 days or over 20 days in comparison to cells that are doubling almost every day under fertile conditions. But if you re-add all of the nutrients and oxygen after 3 weeks under arid conditions, the cells pop right back and they're capable of dividing. So this is not a senescence that the cells are undergoing. So in this setting, Yogev conducted a whole genome screen, whole genome CRISPR screen, A lot of things came out of it. Basically, he was looking for genes that are required under these nutrient-deprived conditions that aren't required under the fertile conditions, asking what vulnerabilities are specific to the nutrient-deprived conditions. So one of the pathways that jumped out as extremely important was the TCA cycle and electron transport chain as really, really critical for these cells under arid conditions. And this was validated also with some small molecules, inhibitor of components of the TCA cycle, or the mitochondrial pyruvate carrier protein. Another thing that came out of the screen genetically and that we validated in a number of ways is that when cells are under these severe nutrient and oxygen deprived conditions, forcing them to start undergoing anabolic activity is itself lethal. So if we have cells that are under arid conditions and we give the cells FBS, which causes them to start dividing, that also causes the cells to die at a much higher rate. This can be rescued by glucose. So if we give cells the building blocks to be able to deal with this now proliferative stress, they no longer die. And this can be reversed by a number of drugs that inhibit that anabolic demand that is caused by the addition of serum. So cell cycle inhibitors or protein synthesis inhibitors. But one of the most interesting things that we found out of the screen was an absolute dependency on a gene called BCL-XL, a member of the BCL2 family. BCL-XL is highly depleted in cells under these arid conditions, which is what the y-axis here is showing. But there's really no depletion at all under fertile conditions. So that's illustrated in these competition experiments where we're mixing BCL-XL knockout cells with wild type cells. The BCL-XL knockout cells are severely depleted under arid conditions, whereas cells under fertile conditions really don't suffer at all if they're missing BCL-XL. There are small molecule inhibitors of the drug, and we can see that under these nutrient-rich conditions, the inhibitor has no effect on cell viability, but cells grown under arid conditions, either acutely or for more than a week or two, are very, very sensitive to this small molecule inhibitor. As I mentioned, BCL-XL is a member of this BCL2 family, and it has two main functions. The one that it's best known for is a regulator of cell survival and apoptosis, but it turns out that these family members are also involved in cell cycle regulation. Specifically, they act as inhibitors of the cell cycle through mechanisms that are not fully understood. So I'm going to jump ahead and just show you that BCL-XL is required selectively in these metabolically deprived tumor cells in vivo. And so what I'm showing you here is if we take a tumor and we treat it with standard chemotherapy, and then we look using this dextran assay, how far away are the tumor cells when they're dying, we find that the standard chemotherapy really only exerts its effect on killing in cells that are close to blood vessels. But if we now add this BCL-XL inhibitor, we see that most of the activity, most of the killing is not actually happening in the cells that are close to the blood vessels. It's happening in the cells that are further away from the blood vessels. And this gave us the idea that perhaps we might be able to target both in some way. The cells that are proliferating because they're close to a blood vessel might be susceptible to standard chemotherapy, whereas those that are in the quiescent state might be more dependent on BCL-XL and subject to killing by that inhibitor. So we did the following experiment where we actually dosed animals with both drugs. Either drug alone had a pretty small effect on tumor growth and really no regressions, but quite remarkably, we saw that tumors regressed when you added this BCL-XL inhibitor to standard chemotherapy. Now, this is likely going to be toxic in this kind of iteration. but at least it's a proof of concept that targeting both the proliferative component and the quiescent component of a tumor may have some therapeutic benefit. And finally, we think that the mechanism for this BCL-XL dependency is both of the characteristics that we found to be important through our screen, which is that BCL-XL is acting as both an inhibitor of cell death and as an inhibitor of cell cycle. So if we look here and we adjust cells from fertile conditions to arid conditions after 1 day, BCL-XL, the absence of BCL-XL leads to much higher rates of cell death. If we do that transition to fertile conditions, we can see that the rate of proliferation drops down in the wild type cells, but BCL-XL knockout cells can't constrain their proliferation anymore, and that's also probably contributing to their death. So summary here, tumors contain both proliferative quiescent subpopulations, and it's actually the quiescent cells that are the majority of the tumor. And if we give chemotherapy to kill the proliferative cells, these quiescent cells are just going to be able to come back. And that's probably one of the reasons for the failures of standard chemotherapy. There's a marked dependency on oxidative phosphorylation and electron transport when there's metabolic deprivation. We don't know whether this is because the cells are now dependent on ATP for survival, and we're looking into that. A cell, to be able to live in these metabolically deprived conditions, has to repress its anabolic activity, and metabolic deprivation prompts a specific and non-redundant dependency on BCL-XL. And so I think I mentioned all the people who did the work as I was going through the talk. I'm sorry that I've run a little bit over, but thank you again so much for the invitation to speak, and hopefully I have time for a few questions.
Great. Okay, you already have some questions in the Q&A here, and I encourage people to type in some more. If Dr. Stanger doesn't get to him, we'll email him the questions as well. So from Bob Weinberg, you have a question. Are the cells that are disseminated early in primary tumor progression the precursors of eventual micrometastatic disease, or are they just dead ends or distractions?
Thank you for that great question, Dr. Weinberg. You're not the first person to ask that, and it is a great question, and we don't know the answer. And it's a hard question to ask because we would need to have some way of following those very rare cells and seeing what they do. We've tried experimentally a couple of ways of doing that. One way we considered was taking mice that have those early disseminated cells and performing a liver transplant into another mouse. Turns out that's really hard to do in a mouse. We've considered other ways and we don't know. So it's entirely possible that those cells are a dead end. In fact, I think they probably are a dead end and they're not the precursors of later metastasis, but we have no way currently of being able to tease that apart.
Okay, and from Rakesh Jain, you have really excellent work. Congratulations. Two questions. First, on the YFP labeled cells, do you see any of them in lymphatics from mice with Pann lesions or any lymph node metastases? And then secondly, do you only see homotypic clusters in the partial AMT cells? Or do you see when these clusters come, are they always cancer cells? Do they have fibroblasts, leukocytes, etc.?
Two great questions. The lymphatic question, we have not really looked. And I just had a student join the lab, a thesis student join the lab, and he's all about lymphatics. So in hopefully 2 years or 3 years, we'll know a lot more about lymphatic spread in this model. And of course, we also plan on looking at human tumor specimens to understand lymphatic spread in human pancreas cancer. Your second question was about the makeup of the clusters. In our model, most of the time, the clusters are small. So that's the first thing I should say. The most common size of a cluster is 2 cells, and they kind of decrease in frequency as you go up from there. We do see clusters that are 10 or 20 cells. Those are relatively less frequent. And we do sometimes, although not that often, see other cell types migrating with the clusters. We've done a little bit of characterization of that. Occasionally, it's a CD45 cell. I don't think we've seen evidence of fibroblasts coming along. We've looked for evidence of platelets coming along because there's been reports of platelets acting as kind of a shield for tumor cell clusters, and we don't see that either. But I don't know whether that's the model that's responsible for that or just that we haven't looked hard enough.
Okay, and from Justin Becker, who says hi, have you explored whether NSD2 H3K36 methylation is also a regulator of developmental EMT or other developmental transitions, or is this something that's unique to the tumor?
Another great question. So I kind of gave a little hint there that our belief and assumption is that cancers don't really do that much that's new. That's not completely true, but most of what they do, they've repurposed from development or from some other physiologic setting. So it would stand to reason that this H3K36 mechanism of EMT in the tumor cells is conserved from something in development. So we've started to address that by using mice that have that mutant H3K36 M mutation, so they can't form that dimethyl mark. And when we make animals that express that mutant that can't undergo dimethylation in embryos, the embryos die at around day 10 or day 12, and they're very malformed. So that at least tells us that we don't think that this is involved in the EMT of gastrulation, because we think that the animals are able to gastrulate normally. But we have a lot of clues that make us think that the neural crest migration might involve this kind of an EMT. For example, this mark is present in neural crest precursors in the neural tube, etc. So we're currently exploring that. It's a little piece of embryology that we brought back into the lab by looking at these embryos and trying to see whether this cancer identified mechanism is actually a developmental mechanism. Great question.
That is really cool. Okay, so you have two questions from Andy Aguirre, who says, "Hi Ben, great talk. The BCL-XL observation's fascinating. Is the protein expression upregulated in the arid regions of the tumor? Can you see this in human specimens by IF or IHC? And then kind of a follow-up, would you say that this quiescent cell state that you describe in the arid regions is a type of cancer cell dormancy?"
Great questions. So I know that Yogev knows the answer to your first questions about expression of BCL-XL. We haven't seen it in human tumors, I think because just we haven't felt comfortable with the antibody reagents. So we haven't really been able to look at that. Probably should try harder. It's a really good question. Your question about, is this a form of dormancy? I think we get into semantics a little bit. I guess you could call it a form of dormancy. We use the term quiescence instead, because I really think of dormancy in more of a clinical setting where you have a cell that's, that has disseminated and is living for some long period of time dormant before it reemerges. And in that setting of dormancy, we don't really necessarily think that the cells are dormant because they're nutrient and metabolically deprived. They're dormant through some other mechanism that we really don't understand because we don't understand dormancy. So you could think of it as a form of dormancy, but it really depends on the terminology that you want to use.
Great. Unfortunately, we have a hard stop at 5:05, so you have a couple more questions that we'll send to Dr. Stenger by email. But thank you very much for this wonderful talk, and we're very excited to have you present today.
Thanks everyone for coming. Be safe.